Thank you for attending. I hope you didn't show up only to be disappointed that you're not hearing from Promothera, but you're hearing from NexImmune. They said, my name is Scott Carmer, and I will be representing NexImmune today. We're a small preclinical company. We're located in Gaithersburg, Maryland. It's about 20 miles outside of Washington, D.C. And we're an immunotherapy company developing a novel technology platform. Like every immunotherapy company focusing on cancer, our goal is to produce cancer antigen-specific T cells. But maybe, unlike some other companies, we think we can do so with what we'll call pharmaceutical precision. And I'll elaborate that throughout the presentation. But at the heart of our technology are artificial antigen presenting cells. And because these are synthetic, we can load these artificial antigen presenting cells with multiple cancer-specific antigens. And we think that this then delivers the ability to, when we stimulate T cells, we see a polyclonal response. So we stimulate multiple clonal sets of T cells. And because we're stimulating them with multiple antigens, we see enhanced cytotoxic activity. So when we look at cytokine expression, we see greater subsets of cytokines being expressed through these multiple clones of T cells. We're developing the technology both for in vivo and in vitro application. Our first product, AIM-101, will be delivered for subcutaneous administration. We will be exploring it in multiple tumor types, both solid and liquid, and we hope to be in the clinic in phase 1 trials early in 2016. In terms of financing, there's been about $20 million invested in the technology to date. Our current investors include NEA, Pfizer Ventures, and Amgen Ventures. We've benefited from multiple corporate and academic grants, both at the federal and state level. And I think, like everyone here, we're actively seeking R&D partnerships. So a bit about the technology specifically. As I mentioned, it's a highly flexible, synthetic approach to cancer antigen-specific immunotherapy. At the heart of it are these synthetic nanobeads. So we take these beads, and throughout our manufacturing process, we covalently link, in this case, a recombinant, fully human HLA IgG4 molecule. In terms of an antigen-presenting cell, this delivers your classic signal 1. And then simultaneously, we covalently link a humanized co-stimulatory molecule, in this instance, anti-CD28. So our final product is an artificial antigen-presenting cell, fully capable of eliciting classic signal 1 and signal 2 for T cell stimulation. We believe that this is an approach that allows us to maximize the utility of our known cancer antigens. So again, we can take individual beads and we can load them with a specific antigen and take a separate bead and load it with a separate antigen. So we can take several different antigens into the body in the same formulation. We think that's what drives this polyclonal response. And the T cell population, through stimulation with these multiple antigens, elicits this enhanced cytotoxic activity. So we think we can maximize the utility of the known antigens, and we think we can maximize the efficacy of antigen-specific T cell stimulation through this technology. We talked about pharmaceutical precision. So if you take a step back, the manufacturing process for this technology is somewhat different than would be associated with cell-based therapeutics. So our first product will be sub-Q administered. We'll produce the product in bulk vials. We can package either in multi-dose vials or in single-unit use subcutaneous injections. And we see very direct distribution, straightforward, which ensures broad access to patients. This might be contrasted perhaps with other cell-based therapies that have limited shelf life and therefore limited distribution. So instead of going to perhaps just 50 or 100 cancer institutions, we see distribution to literally thousands of oncologist offices. Having said all that, we are also developing the product for adoptive transfer for a couple reasons, one of which is We're currently pursuing, through deep gene sequencing, the identification of patient tumor-specific neoantigens. And it's our hope that we can identify those, isolate those, and use those antigens then to load onto our artificial antigen-presenting cells, stimulate T cells ex vivo, expand, and then reintroduce into the patient via adaptive transfer. We have several preclinical publications and a very broad, strong IP patent estate. The technology we've licensed exclusively from Johns Hopkins. So a bit more about the manufacturing process schematically. So again, it starts with this nanobead. In this instance, it's a PLGA PEG biosynthetic 1 to 200 nanometer sphere. We load it with the classical signal 1, signal 2. After that, you can see we can load each individual aAPC with a very specific antigen. Right? We produce these in volume, we mix them together, and again, we package either through multi-dose vials or unit-of-use syringes, and then we ship directly to physicians. So in terms of the process itself, from a QC, from a final kind of release assay perspective, we would anticipate very minimal batch-to-batch variability, and because we're not using cells, we wouldn't be concerned with patient-to-patient variability. It's a process also we would expect to generate very manageable cost of goods. So I'll go through this fairly quickly. This audience doesn't need an education about the immune system, but in the intact immune system, when we detect cancer cell, T cell becomes stimulated, propagates, it identifies and kills the cancer. When we have malignancies in cancer, cancer cells have found a way to evade or to bypass the immune system, and they do so in a couple ways. One is they can directly downregulate a T cell, so they can just neutralize it, or they actually have the ability to re-engineer a naturally occurring antigen-presenting cell. And so they reprogram it to basically come into contact with a T cell. Instead of upregulating or stimulating it, they downregulate it. So here you have T cells that are where they're supposed to be in the microenvironment, but they're uneducated and they're unstimulated, and so they just sit there. So with the aAPC, the promise that we're moving forward with is that we bypass the tumor's ability to downregulate T cells. Because we're synthetic, we're not exposed to a cancer cell's ability to reprogram the APC. So we find the T cell, we're antigen specific, we educate that T cell to respond to that specific antigen, and then we stimulate or upregulate that T cell. Now we have an educated, fully active, stimulated T cell. It propagates, finds the cancer cells, and kills the cancer cells. Can I advance the slide? No? There we go. So just two quick data slides. This is our proof of concept in the classic preclinical melanoma mouse model. So day 0, these animals were injected with melanoma tumor cells. They migrate, they localize in the lung. Day 3, the mice are injected with low avidity CD8 positive T cells. And then on day 4, 5, and 6, a subset of mice are injected with aAPCs loaded with melanoma peptide. Day 15, the animals are sacrificed, and we look for tumor burden. And in this model, tumor burden is characterized by number of tumors, not by tumor volume. So to orient you to the data, the first bar graph, these are untreated mice, and you can see significant numbers of tumor. Literally, the lungs are peppered with tumors, 250, 275 tumors. The next bar graph represents the tumor cells plus the low avidity CD8 positive T cells, and you see marginal benefit. Bars 3 and 4 represent the artificial antigen presenting cells that were loaded with melanoma peptide. Bar 3 is from one batch, and you can see significant dramatic reduction in the number of tumors. And it was reproduced in a separate batch to show reproducibility from batch to batch and reproducibility of effect. The next bar graph, these animals were injected with aAPCs that were capable of eliciting signal 1 only, and you see marginal benefit, no benefit on tumors. The next bar graph, these were aAPCs that had the capacity to elicit signal 2 only, and you see marginal benefit. And the last bar graph was sham or placebo aAPCs. So I think this demonstrates a couple things. One is that when injected in vivo, These cells have the ability to migrate and find the tumor cells wherever they're localized. And two, when they find them, they have the ability to destroy them. So we talked about polyfunctionality. This was a study that was designed to look at cytokine expression. So here we have human CD8 positive T cells that have been stimulated with peptide-loaded either artificial antigen presenting cells, dendritic cells, or T2 cells. After stimulation, we look and check for cytokines. And here we're looking for the number of different cytokines released, and we're testing for 5 separate cytokines. And the pie charts represent in color the percent of cells that are expressing either 1, 2, 3, 4, or all 5 cytokines. And so the headline is the aAPCs, 32% of those T cells were secreting all 5 cytokines that were measured. The natural dendritic cells, 13% of those cells were expressing all 5 cytokines, and the T2 cells, only 5%. So this enhanced cytokine expression is what we think leads to the greater cytotoxic potential that the aAPCs have. So to kind of summarize, we think it all comes back to the synthetic nature of this approach and the ability then to arm or to load these aAPCs with multiple cancer-specific antigens, each one eliciting a polyclonal or a separate clonal response. So more and different T cells finding and fighting cancer. Each one of these is stimulated T cells delivers enhanced cytotoxic effect through this polyfunctionality, the expression of multiple subsets of cytokines. We talked about the ability of these cells to bypass downregulation within the tumor environment. And unlike naturally occurring aAPCs, these can't be reprogrammed by the tumor to then act on behalf or the benefit of the tumor. We see broad utility for this technology, and we'll be developing it across several tumors, both solid and liquid. And we think that we have the benefit of mass production and pharmaceutical precision. Our milestones coming up, we're just completing our GMP manufacturing process. Once we get the final cells, we'll run our tox studies, submit our IND, and hope to be in the clinic, like I said, in the first half of 2016. 18 to 38 months, we hope to be through phase 1. We'll be testing the clinical utility as monotherapy. In phase 1, we'll also be testing this in combination with checkpoint inhibitors. And in parallel to our phase 1 program, we'll continue to evolve our understanding in the technology, looking at personalized application and the potential to use neoantigens with the aAPCs. Hopefully, we'll have that technology progressed in order to make an informed decision on our phase 2 clinical trial design. And with that, I'll leave you with the list of our management team. And we have been greatly benefited by a wonderful cast of scientific advisors. So with that, I would thank you for your time and go to the next presenter.